Good morning. I'm sorry for stealing the thunder of Alarta with my slide. I'm Gustavo Mahler. I'm the CEO of Cellistic, and I would like to introduce our company. We have been here before. We basically create solutions for off-the-shelf iPSC-based cell therapy in the immune space, and we have done significant progress on offering these capabilities to potential customers. So, as you all know, cell therapies have become possible in the last years. That said, there are significant obstacles to become mainstream. Those obstacles come from cost. We have heard some explanations in the last sessions about the high cost and the issues that has for the distribution of these therapies. Also availability, the complexity of the methodologies for manufacturing cell therapies make availability very limited, and less than 50% of the patients that actually are potential targets for these therapies end up treated. There are some actually papers recently published that talk about even less than 20%. That is a pity given the amount of time and effort that has taken to develop these therapies. There are some questioning about quality and characterization of these products as they are manufactured patient by patient, and off-target editing is very difficult to control. They are difficult to scale with massive capital investments required to scale, and they are lengthy and costly to develop. It actually really takes a lot of time. It has a lot of complexity. The overall clinical development and manufacturing process development, it's indeed one of the most challenging undertakings in our industry. So what we came up with is a solution to all these problems at the same time. Our solution is basically a pre-established platform that enables to manufacture cell therapies the same way you will manufacture antibodies, creating a cell bank first based on immune pluripotent stem cells that are pre-edited. And that cell bank then is used to expand and differentiate into the target cell type. Our platform includes the first part, that is the generation of that cell bank, that is called Pulse. And basically what we do there is generate the iPSCs, generate the edits necessary, and test those cells, and then create a cell bank. And a second part of the platform called Echo, where the cells are actually expanded and differentiated simultaneously, all the way up to the production of the drug product to be able to deliver an off-the-shelf solution of product and much lower cost. We at Cellistic have developed different platforms for different types of cells. They all are based in the same kind of approach, and it's a scalable approach. So let's talk a bit more about how that is done. So if you look at that, there are different points of entry for a customer. A client could actually ask us to start with a donor cell and create iPSCs ourselves. Those iPSCs we create can be edited using our proprietary STAR CRISPR program. And then those cells, after doing the editing, including cloaking to avoid immune response, those can be then introduced into a GMP facility and banked. GMP processes start all the way from reprogramming up to the manufacturing of the cell bank, ensuring that all the cells are produced at the GMP under EMA and FDA requirements. Clients could decide to enter at the stage of the donor, at the stage of bringing their own iPSC bank for editing, at the stage of taking some pre-edited iPSC banks that we have that actually have some of the cloaking already included, or they can bring their own cell bank generated somewhere else. Those cell banks are stored, normally cryopreserved, and then they are cracked as you cracked a normal CHO cell vial and expanded through a manufacturing process while the cells are differentiated and simultaneously being expanded, creating then a drug product that can be then labeled, cryopreserved, and distributed. So if we talk about the first part of the process, that is pulse, there are certain characteristics of what we have built in that platform that are relevant to ensure that when customers start with us, they actually carry all the relevant and compliant aspects, and including intellectual property aspect, into consideration when they develop their products. If you look at, for example, the capability of starting from a cell and then getting to edit, we have obviously reprogramming technology, and we have also our own CRISPR technology, although we can work also with MAT7 or Cas9. And all the process is done end-to-end under GMP principles, including the cell banking that is done under EMA or FDA GMP. We also have a clear IP position on all these. We have the rights for reprogramming and for the editing. Therefore, customers take one license from us for their cell line that covers all the concepts, all the way from development, all the way to commercialization, with clear economics, a clear protection in all the way. In addition to that, we have very interesting capacity. We have been able to automate our processes to reduce the time consumption of this process, as well as the cost, and we have very fine-tuned procedures that ensure very high likelihood of success. At this point in time, our likelihood of success creating edited cell lines is over 90%. And we obviously have a dedicated quality system that has been audited by the EMA and is compliant with the EMA and FDA standards that include all the usual requirements for GMP, for the manufacturing of cell lines. In addition to that, we have launched, beginning of this year, a pre-made iPSC cell line that includes edits that allow for cloaking. Basically, two of the edits, one for HLA-A and HLA-B knockout, are already included in this cell line that is already fully tested and available for customers to start the editing. So customer could come, we have the cell line available, can bring their car, we can easily integrate by knocking in that car. We can do up to 3 knock-ins in one round of editing, and then we can create then the cell line of interest for the customer in a much shorter time. The advantages are obviously all the IP is under one roof, excluding obviously the IP that the customer brings in. All is done under GMP standards. We guarantee that these cells will indeed differentiate into the cell type of interest, and we have extensive documentation of our cell line from the donor all the way to the end of the cell line. That is something very important at the time you need to file an IND. So if you take into account how long it takes that, normally in general, previous innovators have taken 2 to 3 years to generate a cell line. If we do what we call pulse, that is the lower graph you see in light blue, it takes us around 18 months from a moment we actually start to work with somebody to create a GMP and release a GMP cell bank. Obviously, if somebody starts with our allo chassis, that creates a significant saves of time. So basically, we can actually do that under 1 year all the way through. In addition to that, we offer a standalone, or for any customer that brings their cell line, or for our own work with our own cell lines, the CRISPR editing process. We actually acquired this last year from a company in Colorado. We included all the intellectual property, and CRISPR has a very high efficiency for knock-ins, much higher than other alternative nucleases, has a very good efficiency for knockouts, and it has an impressive performance on off-target editing, much lower than any of the other nucleases currently in the market. And that we offer both for our own work, for the work with our cell lines, with customer cell lines, or a standalone for other applications. Regarding the manufacturing side of the platform, the eco part of the platform, we basically have established a family of platforms, a eco-NK platform, eco-gamma-delta platform, alpha-beta platform, and a macrophage platform. Those are actually all designed with affordability in mind, ensuring that we minimize cost and maximize scale. All the steps are actually done in a way that can be scaled in bioreactors, is one of the secrets, and achieve up to 100 liters of scale. All the manufacturing is actually geared towards creating a product that is off the shelf and ready to go for the patient. We are fully compliant with EMA and FDA requirements for GMP. We have our own facility that it directly is prepared and designed to run our own processes. And we actually did have capacity in that facility to run multiple processes and products, and it's operated next door to our development labs. Therefore, there is easy transfer of processes between the process development and the manufacturing. It's important to understand, a lot of people claim they have processes. We actually can show you the processes. It's actually the real thing. This actually is our eco platform. Here you see the different unit operations. The first 3 unit operations, where we start from a vial, as I mentioned before. We expand first into small T-flask, then through another expansion surface, and then we finish in bioreactors to produce hematopoietic stem cells that can be directly used or frozen down for multiple uses later on. Then the unit operations 4 continue with the differentiation from HSCs into NKs, and we finish with the last unit operation 5 in the manufacturing side, where we scale up the process for NKs in this case. Then we proceed with the fill finish of the product, and the product is then cryopreserved. The entire platform, end-to-end, has been already run at manufacturing scale. We have created batches of 10 liters with 22 billion cells per batch. That is a pretty high yield. All the process has very low COGS, is feeder-free. We don't have feeder cell lines included in the process. All the materials have been qualified under GMP, and the process has established and transferred to manufacturing indeed, and is scalable to up to 100 liters. In the graph, in the lower right side, you can see a comparison of cost, assuming a concentration of 100 million cells per dose, of what a dose of NK treatment will be, the cost of a dose of NK treatment will be for an autologous process, for an donor-derived allogeneic process, and for Cellistic process. We actually are able to develop a process that yields cost of goods under $50,000 per treatment, and sometimes even lower, as we have demonstrated by one of our partners where we are working currently on. We have also advanced our alpha-beta T-cell platform. Here, the first 3 steps seem recognizable, are the same for producing the hematopoietic stem cells. The last unit operations 4 and 5 are slightly different because we actually go directly into bioreactors in suspension, and from there we do to fill finish and cryopreservation. Our process, again, is feeder-free, is all chemically defined medium, is scalable, and we actually can, by adjusting the characteristics of the last 2 unit operations, 4 and 5, we can adjust if we obtain CD4 or CD8 cells. And you see that in the graphs on the lower right, that those CD4 and CD8 cells can be actually produced in large volumes without actually any segregation methods. And the profile of those CD4 cells is indistinguishable from the profile of native CD4 cells. Last, we also have a partnership specifically for the production of Tregs with our partner Quell. We have been using a similar approach, created a complete process where we can indeed produce Tregs that actually in functional assays as flow profiling, epigenetic profiling, and Treg activation profile, actually they match native Tregs, and this is a pioneer biology. We believe we are the first ones producing large-scale Tregs from iPSCs. So in summary, we have a iPSC-based platform to provide cost-effective, scalable solution for the development of immune cell therapies, right? So this creates significant benefits of economy of scale as you scale up and not scale out. It's a reliable and consistent quality product. The product becomes available for the patient because it's off the shelf, and we can actually expedite product development due to the fact that most of these unit operations, as well as the cell lines, are platformed. And the allo chassis that we presented and some of the ready-made iPSC cell lines allow for customers to immediately start development in a compliant, fast way and actually accelerate the CMC section of the development of a cell therapy with a fully documented operation under GMP and with really fast timelines and an affordable cost. Thank you very much, and thank you for the opportunity to be in here. Goodbye.